bigger and bigger. And so today we're going to talk about some stuff that is not even considered living. We don't even talk about it as a living thing because the definition of a living thing is that it has to be able to either reproduce or replicate on its own, and these things cannot do that. So we'll call these almost living things. Okay, so in order to be living, all living things must be able to reproduce or replicate. On their own. Okay, so they have to be able to do it on their own, meaning they aren't going to use anything else, any other organism or anything like that. Okay, and so it's for this reason that this replicate thing comes in handy, because let's talk about humans. Can humans reproduce on their own? No. But can we replicate on our own? Yes, all of our cells are constantly going through mitosis and making more cells, right? But I can't make another human by myself, right? That's the whole sexual reproduction thing. That doesn't work. So being able to replicate on your own means that you're still living. So what are these things that are non-living, but they're really close to living? Well, there's three of them that we're going to talk about today. We're going to start out with the simplest of all of them, and those are called prions. Okay, prions are proteins that had the ability to be folded correctly, but they just got misfolded somewhere along the way. So these are misfolded proteins. Okay, and that in itself is not very scary that they're misfolded. The scary part about them is that these misfolded proteins have the ability to take healthy proteins or properly folded proteins and cause them to be misfolded in the same way that they are. So they essentially like corrupt other proteins, right? So misfolded proteins that can cause healthy proteins to become misfolded. All right, so what's up? Yes, that's what I was just about to talk about. So the most famous version of a prion is called Bovine Spongiform Encephalitis, which is mad cow disease. And basically it's called mad cow disease because their brains get all like punched holes in them, right? So spongiform means that it like turns it into makes it look like a sponge, right? And encephalitis is like the brain, right? So it's a disease of the brain. So basically these prions, they go in and they take some structural proteins that should make up the brain, and they misfold them, right? They take them and they change them into a different shape, and because they're changed in a different shape, then they don't do what they should do, which is provide structure to the brain, and the brain just becomes mush, right? And the cows go crazy and die. Okay, so the question becomes, how does one cow get a prion in the first place? And the answer to that is relatively unknown. We don't know why these misfoldings happen, right? But randomly, we can assume that they will spontaneously generate. That it came from some protein that tried to fold correctly and then got misfolded randomly. Okay. Back about 15 years ago, there was an outbreak of bovine spongiform encephalitis, where in Europe there were entire herds of cows that had to be destroyed, right? Because they got mad cow disease, and they weren't sure if which ones actually had it. And the question becomes, why did a bunch of cows get it if it's this random mutation and it's not really contagious, right? The only way that it could go from one cow to another cow is if that cow's brain part of that cow's brain actually got into the brain of another cow. So how did that happen? How did an entire herd of cows get infected with it? And the answer to that is far worse than you think. So when they slaughter a cow, there's all kinds of parts that are good, right? That they use, right? So they use like the bones of the cow. They grind them down, and they can use them for various byproducts. They can use all the meat from the cow, so all the like muscles and stuff are obviously going to get turned into meat, right? Cow tail is often used in certain recipes in different cultures, so they can even use the tails, right? Cow tongue is a delicacy, but there's certain parts like eyeballs and brains and certain organs that they don't really get used for anything. They don't have any real human use, and so those are just they go into like the junk pile. Like, we don't need this stuff. They leave them out. Okay, so these farmers are like, well, this seems like a waste. Look at all this good meat over here that we have from like the cow intestines and the brain and the eyeballs and stuff. Like, we're not even using that. So what do? They throw it in the meat grinder, and then they put it in the cow's feed, right? So they feed the cow parts, leftover parts, back to the cows, which is all well and good until one of them got bovine spongiform encephalitis, and then you fed that back to the entire herd, and then the entire herd got bovine spongiform encephalitis, and then you had to destroy your entire herd. So now we have laws against that. You cannot feed livestock the same species. Like, so if you get like byproducts from cow, you can't feed them to cows, you have to feed them to chickens. And if you get byproducts from chickens, you can't feed them to chickens, you have to feed them to cows. And that prevents the spread of things like bovine spongiform encephalitis. And previous to this, we didn't have those laws because we didn't know that was a thing. We didn't know that was a real concern. But now we do. And so that's a law. What's up?
So that's stuck to just one species?
Yeah, they're pretty, since the proteins are unique to each species, generally what we find is that the misfolded protein can only misfold proteins of the exact same type, right? And there's not a whole lot of evidence of it going back and forth or between different species. Now, there's certain cases where species share the same proteins, and if it was one of those, then that would be a problem. But like feeding it between a mammal and a bird, as far as chicken to cow, is much less likely to cause a problem. But, you know, if you think about cows are mammals, right? And so are humans. So we're much more closely related to cows than birds are to cows, and so we're much more likely to be able to contract bovine spongiform encephalitis. There's a human version of spongiform encephalitis that it works in almost the exact same way, and pretty much the only time that we see cases of that is when there's like a tribe that is cannibalistic, that like they eat their dead for usually religious, spiritual purposes. Like they think that it allows them their spirit to live on with the tribe. And they can pass on things like prion diseases. Another really scary thing about prion diseases is that we've learned recently that the techniques that we use to sterilize surgical equipment don't destroy prions. So one of the most common ways that we sterilize surgical equipment is by putting it in this thing called an autoclave, and an autoclave increases the pressure and the temperature, and it basically kills everything off using superheated steam. And it's like super good at destroying viruses and bacteria, because there's a lot of things in there that won't be able to deal with the heat. But a lot of prions are folded in the most efficient way to allow them to be the most stable. And so when we put them in the autoclave, they don't denature. They just remain in their prion form. And so you could pass it from one patient to another patient if they had a prion disease. The good news is there's not that many prion diseases, and so it's very unlikely that the person who was on the operating table before you had a prion disease. And also, it's a good thing that a lot of the surgical instruments that we use these days are disposable. We only use them once. So like scalpels are not getting washed and reused. They're getting destroyed. They're disposable. So I wouldn't worry too much about it, but it's interesting. All right, let's talk about viroids. Viroids are pretty rare. Basically, these things are like viruses, except for only the genome part of it, only the RNA portion of the virus, and all it does is reproduce, right? So this is a naked RNA viral genome that does not code for proteins. And only replicates itself. So this thing seems silly to me when I think about it, right? Because all it does is make more copies of itself. That's it. Like, it just goes into bacteria and then makes more copies of it. And that's it. That's its entire life. It doesn't make any proteins or do anything or even have a metabolism or anything. It just makes more of itself, right? But then, when you think about like the biological meaning of life, which is to make more copies of itself, this is like the purest form of that, right? All it doesn't do anything but copy itself. That's just its entire purpose in life. Make copies. Seems like an okay strategy. These are pretty rare. They're pretty rare because they only infect bacteria, because bacteria are the only crazy things that just find random RNA and DNA in the environment and then pull it into the cell and say, I wonder what this does, and then start producing those proteins. A human cell would never do that. The human cell would never just pull in random RNA and then start producing it, because it would likely lead to a disease. Thanks.
No problem. Have a good day.
You too. He doesn't go here anymore.
Oh, really?
Yeah.
All right. Thank you very much.
Yep. All right. Viruses. You guys have heard of viruses, I'm certain, right? These are RNA or DNA genomes that are surrounded by a protective protein shell. Okay, RNA or DNA genomes surrounded by a protective protein shell called a capsid. Okay, so this RNA or DNA that they have codes for a whole bunch of stuff, right? Not only does it tell the host cell, the cell that it infects, to make more copies of the RNA or DNA, but it also tells it to make more of the proteins, right? And so you've probably seen a picture of a virus in a book or something like that. They're often shown, well, that was pretty ugly. They're often shown like this. You guys seen those before? Yes? Okay. This is called a bacteriophage. Okay? And a bacteriophage only infects bacteria. Okay, so this is not what an animal virus would look like, right? And they're much smaller than animal viruses, and they have two parts to them. Okay, they've got in here, this is their viral genome. And here is the protein capsid. This is in contrast to an animal virus. Right? Animal viruses, they have an extra piece to them. So here's the protein capsid. Here's the genome, right? And then they also have a plasma membrane that surrounds them. And this plasma membrane has a combination of markers on the outside of it. Some of them are there to trick your cells into allowing them to get into your body and infect you, right? And the other ones are stolen from your particular cells, because if you remember, the markers that are on the plasma membrane are the glycoproteins and the glycolipids, and anytime something gets in your body that doesn't have the name tags, the glycoproteins and glycolipids that match you, your immune system destroys it. But viruses, they steal those name tags from the cells that they come from, their host cells, and so it tricks your immune system. Your immune system goes, hey, where are you from? And he goes, I belong here. And he checks it and he goes, oh, hey, you're right. Okay, carry on, right? Very like the Force, this is not the virus that you're looking for type of thing, right? Star Wars beds. All right, so what are the parts here? We've got a plasma membrane with protein markers on it, or glycoprotein markers on it. We've got a protein capsid, and in here we've got the viral genome. Okay, so there are 3 cycles that a bacteria can be, or it's not a bacteria, a virus can be in. The first one is called the Lytic Cycle. Okay, these are referred to sometimes as infection cycles, right? So basically, if you had, we'll do bacteria here. So here's a bacteria, here's that bacterial genome, right? And here is some bacteriophage latched on. And it's going to inject its viral genome into the bacteria. This is called infection. The next stage, you now have this viral genome. This is the bacterial genome over here. Bacterial chromosome, and this is the viral genome floating around out there. Nothing's happening yet. This is called incubation. Then this thing starts going to work, and the bacteria starts producing whatever's in the viral genome, right? Sometimes there are specific proteins in the viral genome that disable the bacteria and stop it from doing anything else other than producing the virus, right? So you've got your bacterial chromosome still. You've got your viral genome. Oopsies. And this viral genome, it's just making more copies of the virus over and over again. So viral parts start building up inside here. Lots and lots of viral parts. Makes copies of the genome too. So now we have a whole bunch of little viral parts in there. And then this part is called replication. And then what happens next is an enzyme is released that causes that cell to burst open, and a whole bunch of viruses are released from that bacterial cell. And then they go on to infect other cells. This part is called lysis. Okay, that's a lytic cycle. all viruses will eventually go into the lytic cycle. This has to happen at some point, right? However, sometimes that's not the most efficient way to infect the most cells and produce the most viruses. So there's also something that's called the Lysogenic Cycle. In the Lysogenic Cycle, what happens is infection. So same thing as before. But now, instead of this thing just hanging out next to the genome, it actually gets taken into the bacterial chromosome. So now, this bacterial chromosome gets the viral DNA inserted into it. Right? So this is the viral DNA. And this is the bacterial DNA. And once it's been inserted here, we refer to the viral DNA as a prophage. And it can just lay dormant in there. Okay? We have thousands and thousands and thousands of dormant viruses in our genome. Some of them are millions of years old, right? That just don't get expressed ever. They got inserted as a prophage, and now they're just hanging out, right? So, what's this thing going to do now? What do all bacteria do? They replicate. That's their main goal in life, right? Is to just make more. So this thing is going to go through binary fission. And when this goes through binary fission, guess what? Now both of these cells contain the viral DNA. So basically, now this virus has infected two cells without ever having to make more viruses, right? Now, this can sometimes then go and start replicating, right? And go into a lytic cycle and bursts open, right? And sometimes it's just one cell, and sometimes it's the whole colony that does it at the exact same time, right? And would release millions and millions and millions of copies of viruses into the environment then to infect other colonies. What's up?
What triggers a virus to choose which cycle?
It's mostly based on which virus it is. Like it's like hard coded into their genome. So in this case, it's really simple for bacteriophages. For a bacteriophage, if the genome is made out of RNA, then all it can do is a lytic cycle. But in a bacteriophage, if its genome is made out of DNA, it can be incorporated.
So only DNA ones would be lysogenic?
For bacteriophages, yes. All right, let's talk about one other thing that happens for animal viruses. This is called viral shedding. Occasionally, this is called budding. Okay, so in viral shedding, it's very similar to a lysogenic cycle, except now we're talking about in animals. So we have a different looking virus there. This is going to be animal viruses. And so we've also got a nucleus in here that's protecting the DNA. So here's all the DNA in the nucleus like so, and here's the viral DNA that's been injected. All right, this is infection. Oops, did I forget to do the thing? This is called incorporation. Okay, so just like before, this is a lysogenic cycle, so it gets incorporated. So now we've got the nucleus with the eukaryotic DNA, and then the viral DNA is in there now. That's incorporation. But whereas in a bacteria like this thing remains dormant completely, in viral shedding in an animal virus, it doesn't remain completely dormant. It's mostly dormant, but it can butt off singular viruses, singular viral particles, right, without triggering a full-scale immune response, right? If you make a whole bunch of viruses and release them, that's going to trigger a full immune response, and the cell is probably going to get killed off. But if you just release like one or two every now and then, that kind of goes under the radar. And it's unlikely that the immune system would be able to figure out where that one particle was from, right? This is called viral shedding. Let's talk about herpes. Herpes simplex A is the most common human virus. Something like 70% of the population has it, of the adult population has it. What's up? Sure. So it's the virus that causes cold sores, and cold sores are when that virus has gone into a lytic phase, right? So basically it's been incorporated, and then something triggers it to go into the lytic phase, and then it blows up a bunch of cells, and that's what gives you a cold sore, right? Down here though, this is showing viral shedding. And so somebody who has herpes that doesn't show any symptoms of it, a lot of the population, even that has herpes, never shows symptoms, never gets cold sores in their entire life, right? But you could still get infected because there are these random cells that are giving off just one or two viral particles at a time, enough that it wouldn't start an immune response and you wouldn't show symptoms, but you could still be producing viral particles, okay? So while it's very rare for somebody to get to transmit the herpes virus when they're not showing symptoms, it is possible based on the fact that viral shedding occurs. Now, you are significantly more likely to get herpes or get a herpes infection from somebody who has an active cold sore or something like that, because those are cells that are actively in lysis, and they are releasing millions and millions and millions and millions and millions of viral particles, whereas somebody who's not showing symptoms is only releasing one or two, and so you'd have to get pretty unlucky to get an infection based on that, okay? But point is that it can happen. All right, let's talk about one last thing. Let's talk about the retroviruses. And this period in 24. Okay. So, retroviruses are viruses that contain RNA as their genome, right? But they use an enzyme that's called reverse transcriptase in order to transfer that RNA back into DNA that can then be incorporated into the host cell's DNA, okay? So these are viruses that have RNA genomes, and the enzyme reverse transcriptase to allow them to produce DNA from RNA. So basically, in a retrovirus, RNA is injected into the cell, right, along with a enzyme called reverse transcriptase, and then the cell produces DNA based on that RNA template using this enzyme reverse transcriptase, and then incorporates the DNA into its genome, okay? The major evolutionary advantage that retroviruses have is the fact that reverse transcriptase is super error prone, right? It makes tons of errors when it's going from RNA back to DNA, okay? And we know that anytime you make an error in DNA, that results in a mutation, right? And so these are extremely rapidly mutating viruses because of the fact that reverse transcriptase makes a bunch of errors, okay? So we can say that reverse transcriptase makes errors when producing DNA. Which leads to rapid mutation in retroviruses. So, influenza, the flu, it's a virus, right? There's a flu shot that you get, right? Or you can get. Has anybody ever gotten the flu shot and then gotten the flu? Yeah? Lots. It happens a lot. 4 years ago, 3 years ago, the flu shot that everybody got contained 3 strains. They usually contain 3 strains of the flu, right? And none of them were the prevalent strains that year, because there's so many different strains of the flu because of the fact that it's a retrovirus and it's constantly mutating, right? That they have to essentially guess which ones are going to be the prevalent strains. They do a whole bunch of statistical analysis in order to do that. But if they guess incorrectly, then getting the flu shot doesn't help you at all, right? Because you get immunized against strains that you aren't really in danger of being infected with, right? And the ones that do become the prevalent strains that year, it's too late at that point for them to remake the vaccines, and so you wouldn't know, right? Or they couldn't know. So basically, if they guess correctly on the 3 strains that they put in there, then you're good. Then it will prevent you from getting the flu. But if they guess incorrectly, then it doesn't help you at all. What's up?
Does it vary, like, between, like, regions and countries? Cities and stuff?
Yes and no. Certainly in regions that don't have a lot of people coming in and out of them, like remote regions, they would have their own sets. But as far as like countries with global travel, no, they're all the same. And the WHO, the World Health Organization, is the one that does the guessing. They tell you like which global strains are going to be prevalent that year, and then the vaccine companies produce vaccines with those strains. It's also the same reason that we don't have a vaccine against HIV, right? Because HIV is a retrovirus, so there's not just one HIV virus, there's hundreds of strains of HIV. And actually this became a problem back in like the late 80s. So in the 80s, HIV was really poorly understood, and there was like a community of people who were HIV infected, whether they were drug users. It was really prevalent in the homosexual community back then. And those 2 communities, so they had a higher than average rate, and one of the things that they did was they came together and they were like, you know what, if we just have our own community of people who are already infected, then we can't infect each other because like we already have it, and so we don't have to worry about it. We can, have sex and it's not a problem. Well, turns out that was not correct, because you could essentially get 2 strains of HIV and then you had double HIV, right, which was twice as bad. And even within a single person, you can be chugging along and be okay, and one of your strains can mutate in your body and reinfect you, and then you have worse HIV, right? So, point is, retroviruses, really difficult to make a vaccine against them because they rapidly mutate, and they rapidly mutate because reverse transcriptase is so error prone. Do we have questions on that? What's up?
This is kind of off topic, but I heard this year they stopped the flu mist.
Yes.
Why is that?
Because there was a chance with the flu mist that you could cause somebody else to get an infection because of the way that the flu mist was being produced. you were not using deactivated viruses completely, and so even though you would have an immune response to it, there was a period of time afterwards where you could give somebody the flu who was immunocompromised by getting the mist. And it was like a really small chance, but there were a whole bunch of studies released last year on it, and so they just stopped producing it. What's up?
For the Prions and like the three